Published in 1859, Darwin's Origin of Species revolutionized the way the human body is perceived. Darwin's book hypothesized that humans, along with every other animal, are a result of thousands of years of natural selection, a process whereby favorable traits are retained and unfavorable traits are discarded. He argued that every aspect of humans, from our hair color to our intelligence, has been determined by this process of selection. Now, 150 years later, new technologies are pushing the envelope in terms of what it means to be human and what it means to be a product of the genes of our parents. Discovered recently by researchers studying bacteria, CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, is one of these technologies promising to revolutionize the process of selection. Using a pre-programmed virus, a targeted strand of RNA is injected into a cell. Defense mechanisms of a cell, chiefly protein CAS9, attack this RNA, matching it up to the already existing host cell's DNA. Then, molecular scissors on CAS9 cut into the host cell's DNA and replace it with the RNA injected into the cell previously. This process allows for fabricated DNA created in a lab to be precisely and accurately inserted into the living DNA of any kind of animal cell, including human cells. This technology rapidly moved from studying bacteria to studying genetic disorders. As of 2016, CRISPR is being researched primarily as a means to cure inherent genetic disorders, such as cystic fibrosis and Huntington's disease. However, since CRISPR is able to target any part of the human genome, it can be used to change much more. Everything about a person can be changed by CRISPR, from their eye color to their gender. If the past is any indicator of future transgressions, a technology so powerful will not be contained within a relatively small niche. Instead, it will be used to edit every part of the human genome. Scientists often set boundaries on the reaches of their technologies. However, they just as frequently violate previously set boundaries. Just recently, the limit on the amount of time a human embryo should be allowed to live outside the womb was shattered. Scientists are dangerously playing like gods, and the rest of the human race might have to suffer for it. CRISPR has the potential to exacerbate two already crippling issues in society. The first of these is the issue of gendercide. Utilized extensively throughout many countries, but especially in India and China, gendercide is the deliberate killing of a child because of his or her sex. In India and China, girls are aborted by their parents, who are often motivated by one-child policies. And although many of these policies have recently been removed, gendercide still continues in almost its full previous capacity. Girls are aborted because of issues with money, caste systems, and family honor. With CRISPR, this practice will be radically increased, but in a slightly different way. CRISPR provides the opportunity to change the sex of an unborn child, and with societal peer pressures to have male children, parents all around the world will change the sex of their children. Aside from the clear ethical violation in changing the sex of one's child, the extreme gender imbalances that will ensue will be catastrophic to the world. The second prevalent issue that will be exacerbated by CRISPR is the cycle of socialization. The cycle of socialization is usually defined as the cycle by which individuals are molded by their parents and by society, and the process by which those individuals then continue the cycle by passing it down to their own children. With CRISPR, this cycle will be extended to the human genome. Although parents already pass down traits to their children, genes outside of a parent's own genome can now be bestowed upon offspring. These children will then feel compelled to do the same to their children, perpetuating a cycle that gains momentum with each new generation. In addition to this, poor people will not be able to afford the luxuries promised by CRISPR, and so will be extremely marginalized and pushed out of a genetically altered society. To understand the ethical dilemma created by CRISPR, we must travel back 300 years to the 18th century and Immanuel Kant. In his ethical theory, he echoed an Aquinian sentiment in stating that the intent of an action matters more so than the result. He called this the goodwill, formally stating that actions must be defensible by their intent, not their result. To understand whether or not the intent of an action is ethically just, he formulated the categorical imperative, the first formulation of which is crucial to understanding CRISPR. The first formulation is as follows: act only according to that maxim whereby you can, at the same time, will that it should become a universal law. In simple terms, this formulation states that an individual should only pursue an action if that action, pursued by everyone else, would not result in chaos. An example Kant gives is dishonesty. If a person borrows money with the intent of not paying it back, he may be safe from consequence. However, if nobody had the intent of paying back money when they borrowed, nobody would lend money, for they knew that it would not be paid back. With this formulation applied, one can see that the use of CRISPR can never become a universal maxim. While restricted use of CRISPR may be beneficial to areas like genetic diseases, widespread use of this technology, or in contradiction, a universal use of this technology, would be disastrous, as it would greatly heighten the earlier mentioned ethical issues. One cannot will that it only be used in select circumstances, as this would violate the categorical imperative. My personal ethics argue against the use of CRISPR from a stance consistent with a comprehensive understanding of cultural volatility and human egotism. When the French Revolution arrived, there was a great debate in the intellectual world as to the ramifications of a complete cultural revolution. The dichotomy of thought between Edmund Burke and Thomas Paine sums up this argument. Burke believed that a complete cultural revolution would tear up the roots of society so carefully formed over hundreds of years, while Paine believed that for progress to occur, radical changes had to be implemented. If we look at the human genome much the same as a well-formed culture, the Burkean conservatism can be applied to the human genome. To radically change something formed so perfectly over millions of years would be comparable with looking at a tree, seeing a leaf out of place, and subsequently ripping up the roots of said tree with the goal of changing the leaf. Human culture is volatile. Trends rise and fall, and to have our genome mirror the volatility in our culture would be catastrophic. Fortunately, not all scientists have given in to the temptations of CRISPR. An international initiative led by Nobel Prize-winning biologist David Baltimore is calling for the world to halt research with human embryos in CRISPR until what is being dealt with can be more thoroughly thought through by the global scientific community. Although this initiative has been accepted by the majority of countries in the world, several countries have ignored it and continued on with research using CRISPR, most notably the United Kingdom. It now rests the duty of the public to shift popular opinion away from these gene editing technologies in order that research money going to technologies like CRISPR might decrease. Human evolution has brought us to the point where we can change our own genes. In a way, evolution has become self-referencing. To now think that we as humans have the foresight needed to control this enormously complex system is blatant hubris. If the society that we know is to persist, all research using CRISPR must be halted.